Hi everyone. So we're moving ahead to chapter 12. And for this chapter, I'm actually going to start with a worksheet. So if you haven't, first download this worksheet. And if you can, print it out. If not, then also follow along by writing out the term as we go through it. This will help you remember how to spell it and also help you be able to link the definition with the actual term. So we're going to start by introducing several terminology that I will be using throughout this chapter. We're going into introduction of genetics. This is actually the fun part where we'll get to explain to you how did you inherit that gene from your parents, and also what is the probability of a couple, if they have a certain disease, to pass that on to their child. And moving forward, even we'll teach you how to look at what we call the pedigree and be able to map out in a family, how does a trait get inherited. So all this is coming with the last two chapters of chapter 12 and chapter 13. Okay, to start, we have to introduce this person called Mendel, and he's actually a scientist that actually figured out how this unit of heredity is actually passed on from one generation to the next. So to start, let's talk about some of the terms that I will keep talking about and going back to, okay? So let's look at your worksheet. The first term we're looking for is called a unit of heredity, inheritance passed from parents to offspring. And you already know this. It's a segment of DNA sequence that gives the instruction to make protein. What is it called? Yes, it's a gene. Okay, so let's spell that out. It's a gene. So a gene is a unit of inheritance, and it is a segment of DNA that give instruction to make a protein. We laid this out very early on in Bio 120 to help you understand the definition of a gene, okay? Now moving on, the next, alteration in the gene. So you changed the gene. What is the term that actually alterate a gene? And it changes the sequence. So we have talked about this. It probably goes from A and T, and all of a sudden the A got misspelled, and now it's a T, and then against it is an A, or instead of A, it becomes a G or a C. So a change in the nucleotide, which changes the DNA sequence. And by doing that, it changes the amino acid and thus the trait. What is the changing of a DNA sequence called? Mutation. Very good. Let's spell that out. Mutation. Okay, moving on. Next, different form of the same gene. This I probably only alluded to in the past chapter. So the same gene actually can have different forms. Okay, this is like the hair color. It's all gene for hair pigment, but there are various different form of that same gene. Okay, and this is called the allele. Let's spell this together. Allele. Okay, allele. Allele is different form of the same gene, and it could be a type of mutation. So when you change the spelling of a gene, now it's a different version of the same gene. So this can be a mutation, or it's just a normal different version of it. Like I said, it can be black or blonde hair, but if you have a mutation where your pigment no longer stays on your hair, albino, then that's a mutation, okay? So the sequence can cause slightly different version of the same gene or trait to code for different version of the same trait. This is called allele. So I probably have a different allele than you for the same gene, which is the hair color. Okay, the hair color gene can have different alleles. Now, I might be able to give you a better kind of illustration, okay? So as you guys have learned in high school biology, you probably had heard of the big A, the small a, and they all reference the same gene, right? So that big A, small a are different allele of the same gene. of same gene. Okay, so let me explain that a little bit further. So for example, you got your chromosome 1 from dad, and you got your chromosome 1 from mom, right? And dad's chromosome says black hair, and mom's chromosome for chromosome 1 also have the same gene for hair color, but mom says blonde, and actually they are then called allele of the same gene because they're same for the hair color, but they are of different flavor or variety. Okay, so this is called allele. Okay, now let's move on. Next one. This term refers to the particular gene and allele a person has, their genetic content. Okay, this is called the genotype. Genotype. So I will be using this term quite a bit in this chapter, genotype. That means the kind of genetic makeup that a person has. Now let's think about this. Okay, I'm going to move up a little bit. Okay, what is genotype? Well, let's come back to what I have just illustrated with the big A and small a. Okay, if you have big B from dad, small b from mom, what is your genotype? Well, your genotype is the genetic makeup. Your genetic makeup is then from two different alleles. That is your genetic makeup. You actually have one gene that says put black pigment, another gene that says not so much, so you appear more blonde. That's your genotype. But then what is next? Let's look at this. What kind of trait do you get from this genotype? Well, that's called number 5. The observable trait or characteristic that result from a person's genotype. Okay, so what do you get? What do you think this person's hair color is going to be? Okay, a lot of times the darker pigment is going to trump the lighter pigment. So let's say this is going to be kind of a brunette or a black. So let's go with more simple understanding instead of a middle ground. We'll just say because the black pigment trumps the blonde, having put on more black pigment, you're going to have a trait called black. Then this is called your genotype, which is your genetic makeup. And what you're seeing then is called the phenotype. So let's write that down together on your worksheet. Phenotype. Okay, so the most important thing is right here. Genotype determines phenotype. Whatever genotype you have is going to then dictate what kind of phenotype you have. So if you've done high school biology, then you will know if you have both mom and dad says black, that's your genotype. Your phenotype will then be black hair. Dad says black, mom says blonde, but black trumps blonde by putting on black pigment. So what if the blonde says blonde? You're not going to see it. Then it's black again. Now, what if mom and dad both have blonde? Then you're going to get blonde. So, okay, so these are called genotype, and the trait is called phenotype. Now, let's move on. The next term is called allele or trait, which masks any recessive allele. So again, allele is that different flavor of the same gene. So it can be black or it can be blonde. If you are an allele that masks other alleles, we call you the dominant allele. So dominant is the kind of gene allele that actually masks other type of allele. Okay, so in our example, that will be black. The big B allele is going to mask the small b allele in its trait. And so when you have two copies of the same allele, when you need two copies of the same allele, before you see its trait, before you ever see blonde, then this kind of allele is called recessive allele. Okay. Now, if you need both pair of, if you have both pair of allele identical, so I'm moving on to 8, you need both allele, okay, like I have big B, so dad and mom both gave me the same flavor, the same allele for that gene, then you are called homozygous. Okay, that's Z-Y-G-O-U-S, homozygous. Now, let's try this, okay? If my genotype, which is my genetic makeup, is big B, what is my phenotype? Yes, my phenotype will be black. And how do I describe the alleles I have? Well, they are called homozygous because they're the same. Well, if my genotype is little b, they're recessive, they're also homozygous because they're both allele are identical. Then my phenotype is what the little b generates, which is blonde, okay? So you'll need to be able to interchange these terminologies. Genotype, the genetic makeup, like this. Phenotype, what you actually see in trait. Homozygous is describing the genotype you have. If they're the same allele, they're called homozygous. If there are different alleles, then they are called heterozygous. Okay. Okay, so let's put it here. Heterozygous. Okay, so up until now, the goal is you'll be able to distinguish these terms, okay? What is a gene? When you have a change, it's called mutation. When you have different flavor of the same gene, they're all considered allele for that gene. And whatever two alleles you put together, that is the genotype this individual has for that gene. And that genotype is going to determine the kind of trait that is shown, which is the phenotype. And two alleles, one might mask the other one, that's the dominant one. And if you need two of the same before you see the trait, then that's called recessive, because they both are lower in their voice and you need both of them until it's heard. Now, if you have allele that are identical, you're homozygous. If you are different, big B, small b, you're called heterozygous. Okay, moving along, the last one, okay, is a situation where offspring inherit a pair of identical allele for a trait generation after generation. That means every generation it passed on, it's always the same allele, okay? And we'll probably be using this later on. This is called a true bred, okay? Okay, so in the future, when I said this is a true bred or a purebred, then I am referring to the fact that they have identical allele from generation to generation. You'll probably read this on the textbook, so it helps you kind of identify what it means. Now, let's move on to Mendel's law, okay? And this kind of refers actually back to what we have learned in mitosis and meiosis and how a gene is actually inherited, because we have to go back to meiosis because that's where the sex cells are made, okay? And when Mendel did this, he hasn't learned meiosis, he doesn't know. He was trying to figure out how is that unit of inheritance work. The interesting thing is he's actually a really good mathematician, and he figured out by planting peas that how this genetics works. actually is not just a mixing of blood. That's not how it works, he found out. It's not if I am tall and I mix with someone that's short, then I would get somewhere in the middle. He found something very interesting. Okay. He found that when he have a tall pea plant, so that is dominant, and he crossed that tall with a tall, he gets tall. Okay, that's fine. But if he keep crossing and make sure it's all true bred and keep that tall, and he does the short with the short and keep doing that every generation to make sure that they are all pure. There's no other trait. Okay, it's all short. So now by now he's like, okay, good. These two have only the purest gene and I crossed them. Guess what he found? First, the purebred tall and the short, he ended up finding it's all tall, all of them. So that was puzzling to him. Where did the short go? So that was his first generation. Okay, we call it F1. I am mainly talking about the phenotype, the trait he was seeing. When he would cross all the tall with the short, phenotypically short, and they're purebreds, he realized he's getting all talls. So that was puzzling because he didn't know where the short went. Now, when he crossed all the F1, the first generation, with the first generation, mind you, they're all tall, right? So when he crossed the tall with the tall, but from the first generation, he got something surprising. He actually got some tall and some short. Okay, and that's what he found out. And he was very puzzled by this because when he had a parent that was short and a parent that was tall, everything turned out tall. And then now when he crossed that generation, which are all tall, self-crossed them, then he got tall and short. He's like, what's going on? And then so he counted how many of this, and then he found out it had a specific ratio. It was 3-to-1. And by being very good at math, he found out that the only way this can be is a lot like computer. Your genes talk in binary. That means it has 2 codes. And these codes talk just like your computer. Well, he didn't know computer by then, but he was good at math. He's like, you know what? He established these laws. He's like, I know the only way to get 3-to-1 is for every gene, you have 2 says. Okay? He didn't know about homologous chromosome, but this is what he listed. So let's go into his laws. The first law he has is called law of segregation. He realized that for every trait, okay, there are how many alleles? So for every gene, that is as well. You have 2 allele, as we've talked about, but he figured this out through math. He's like, the only way I can get that 3-to-1 is for this trait of being tall. I have each pea plant have 2 alleles that contribute to the final trait. Okay, so each individual has 2 alleles for each trait. And he said, guess what happened during meiosis or when this was passed out? He said the allele actually separates during formation of the gametes. Okay, so if I am an individual, he is saying that for my hair color, I got 2 say, okay, number 1, number 2. And when I form my gametes, which is my egg, they will separate. And I only get this or I get this. And each gamete, so whether it's my egg or a male sperm, that's the gamete, contain how many allele per trait? Do you get 2? Remember meiosis, you get half of everything. So you get 1 allele per trait in your gamete, right? So I put big B in my egg, I put little b in my egg, and potentially my partner, for example, if he has blonde hair, then he puts little b in his sperm. So Mendel figured out that for each unit of inheritance, there's 2 says per individual, and it actually separates during gamete formation. This is called law of segregation. It separates. Allele of same gene separates. And when you fertilize to give new individual, then how many alleles do you have per trait? Let's see. If I married him and have kids, 1 egg meet with 1 sperm, I get the baby. There you go. You get 2 allele per trait. So he knew this before he knew any of the meiosis, but he figured this out through math. He said, this is the only way that my pea plants can have tall with short, all tall, and all tall F1 generation, the first generation, can then self-cross and get tall and short. So my short skipped generation, but showed up again. He said, this is the only way it's going to work. And I'll show you how this works in Punnett square very soon. Okay, so what is law of segregation? Law of segregation is describing the behavior from allele of same gene. Okay, so law of segregation, the first law, is describing how allele from the same gene works. Big B, small b are both allele of your hair color. They are allele of the same gene. And how do they behave? They behave by segregating or separating during meiosis or during the formation of the gametes. That's law 1. Let's talk about what he figured out for the second law. The second law, let's move up. Okay. What he figured out is the second law is called law of independent assortment. What is this law? Well, this is telling us about each pair of allele. What is each pair of allele? That means I have a pair of allele here for my hair color, okay? And potentially I have another pair of allele here for another trait, let's say my eye color. How do they behave? If I get black hair, do I always get darker eyes? For example, big A stands for brown eyes. Okay, is that always the case? And law of independent assortment is saying, nope, they sort independently. If I get big B, I have equal chance of getting big A or little a. So law of independent assortment says each pair of allele segregates independently from the other pair. So this one, big B, small b, is going to sort on its own, and big A, little a can sort on its own as well, which means I have equal chance of getting big B with big A or big B with small b with big A, or small b with little a. I have equal chance to come into the same gametes, okay? And so that will be my example, is that the two set of pairs of allele can sort independently. If I am heterozygous in my hair color and heterozygous in my eye color, okay? Remember, when I make a gametes, I get half of everything. I don't get both gametes. I get either or. I get the big B or the small b. I get the big A or the small a. But the truth is, when I get big B, I got equal chance of getting A or small a. So each of this possibility is 1/4. And we'll talk more about that moving on. Okay. So that is just how gene is inherited. It doesn't distinguish between pairs. They have equal chance of mismatching. And we have already talked about this back in meiosis. Okay, think back. When did we get all this? So the truth is, all possible combination can occur in the gametes of alleles. So any combination, big B, big A, small b, big A, big B, small b, big A, are all possible. I can generate any kind of these eggs, okay? There's no preference of, if I have dominant, then I'm always matched with dominant. Okay, now let's talk about what incidents that you have learned in meiosis can actually contribute to the first law. What is the first law again? Right, law of segregation, that big B and small b, allele of the same gene, will go into separate gametes. I don't keep them in the same gametes. Do you remember when this happens? Well, let's think back. Where would they live? If I get chromosome 1 from mom or dad, and I get chromosome 1 from mom, remember how they are called homologous chromosome, and they're very, very similar. That means they carry the same gene, but different flavors. So for example, chromosome 1, both of them carry the gene for your hair color. And let's say the chromosome mom gave you is blonde, so little b, and dad gave you is big B. Let's say that's what's going on. So you are a heterozygous, and during meiosis 1, remember, homologous chromosome splits. So you either get this big one or this little one, and that happens during anaphase of meiosis 1, where I know this is what happens, right? Usually, because you have synthesized too, but so it's still the same. And we can talk about crossing over later, but remember, homologous chromosome splits during meiosis 1. If not, go back and check your drawing of meiosis 1. Okay, and that's when this goes into one daughter cell that goes into the other daughter cell. So allele of the same gene actually segregates. Let's write this. During meiosis 1, okay, homologous chromosomes separate. And that's when you got allele of the same gene separates, okay? So what I'm doing here is trying to relate the law of Mendel back to what you have already learned in meiosis. Now let's talk about the second law. What is the second law talking about? If the first law is talking about allele of the same gene, the second law is about allele of different gene, right? So we're talking about how do they behave? Well, let's recall again. These are two different genes. How do they behave? That's what the second law is talking about. Second law talk about, this is gene 1, this is gene 2. Okay, do I inherit them? If I get big B, I always get big A? No. So it says you inherit them independently. Okay, so let's pretend, right? We got 2 chromosomes. Let's say chromosome 1 and chromosome 2. And this is in the individual, okay? And we say, well, this is the hair color that lives on chromosome 1, and then you got another trait that lives on chromosome 2. Now, what happens is there are 2 possible things that happens that can contribute to independent assortment. One is when you line up these during metaphase, your B lines up with your big A. Your big B lines up with your big A. And your small a lines up with your small b. Okay. And obviously, you got 2 of everything, right? And then what happens is you split them, right? So there you go. You end up with little b with little a, or big B with big A. That's one possibility. But we also said during metaphase, you can have various ways of lining up. Okay, there's another way. What's the other way of lining up? Well, you can have big B line up with big A. And you can have small b line up with small a. Okay, so that is how you can end up with, okay, sorry, I probably listed this wrong. This is actually small a. And this is big A. It's hard to talk and draw at the same time, right? This is big A, small a. Okay, so when you segregate, so again, I need to draw 2 because I'm going through meiosis, and you got 2 of everything during meiosis. Okay, so when you segregate during meiosis, what do you get? Now you get little b with big A and big B with small a. We talked about this during meiosis, where this is called independent assortment, that it's not always daddy's chromosome matches up with daddy's chromosome. So it's not always blue with blue on the right or left, and then they all segregate. You can have all kinds of assortment. And so that can contribute to how allele of same gene segregates, and they are independent. They don't always come together, right? Okay, so what will be the incidence? Well, one incident is how chromosome assort independently, or chromosome assort independently. And this happens in metaphase. Okay, remember metaphase? They line up in the middle, but it's not all the red in the left or all the blue in the right. They can align, and you got 23 pairs. So you got all kinds of possibility. Now, what's another possibility? Remember crossing over? That's happening in prophase 1, and that also contributes to how allele of different genes assort. So let's look at that. So let's say now, instead of the gene being on different chromosome, which is actually easier to understand, let's look at what if the gene is on the same chromosome. So let's draw this out. Remember, after synthesis phase, I got 2 of everything. So I'm drawing out 2 of everything. And then let's say I started out with big B, and big A. Okay? So what if the gene is on the same chromosome? Aren't they stuck? Don't I get big B and I would get big A? Don't I always have big B with big A. How did you say you sort them independently? I thought during during meiosis, then, right? Then I would get big B always with big A and little b always with little a. How would I ever get the independent assortment of big B with small a? How can I get that? And how can I get little b with big A? I don't see it. That's when crossing over introduces that opportunity. So remember what happens in crossing over. You got the tetrad lining up together in prophase 1, and you can have non-sister chromatids exchange genetic material. So let's draw that out. What happens is you got tetrad where homologous chromosome can line up and actually exchange genetic information between the two. So then you end up with something like this, okay? Oops. You end up with something like, now they're going to exchange. This blue is going to become red down here, and the red is now going to have blue down here. Now, what does that mean? That means originally, remember what this is? This was big B, correct? Blue. And this bottom was supposed to be big A. But now that I have switched it to red, you see, this is now little a right here. But this one is still big B with big A. And this guy is going to become small b with small a. But this particular one will be, oh, let me get that back, will be small b with this blue, which is what? Big A. Okay. So when you have crossing over, then you will get a gamete. So let's draw this back out. This particular one will be B little a. And that is how you get the big B with little a. This one will be big B with big A. Okay. Big B with big A. So I get that one, I get this one. This one will be small b with small a. That's a sister chromatid, small b with small a. And then hiding behind it will be small b with big A. Small b with big A. So I get all 4 after crossing over, okay? So by understanding meiosis and the specific incidents that happen during meiosis, we can now explain both Mendel law of segregation, and law of independent assortment. So let me kind of conclude. Law of segregation, the first law is talking about allele of same gene. Big B, small b, they're allele of the same gene. What do? Segregate. They separate during gamete formation. What about second law, law of independent assortment? It's talking about allele of different gene. Big B, small b, big A, big, small a. How do they behave? They behave independently. If I have big B, I have equal chance of getting big A or small a. How do I get that opportunity? Through chromosome assort independently during metaphase, they line up randomly, and through crossing over during prophase. Even if you're the gene in the same chromosome, you have equal chance of shuffling with your non-sister chromatids. Okay, so I'm going to stop here, and we're going to make another video moving on for now really applying these laws into Punnett squares.